We were given two proteins of clip P. Protein A is a wild type and protein B is a mutant type. It forms a heptadimer complex where it is formed by 14 subunits where they are assembled to form a barrel-shaped structure. It coordinates with clip A protein which acts as unfoldage and helps in denaturation so that it can easily be cleaved by clip P. Both the proteins are expressed in E. coli. Clip P is a serine protease belonging to the serine peptidase family. It is an ATP dependent and it cleaves proteins such as casein and albumin. The main functions of clip P are it is a chymotrypsin like activity. It helps in the degradation of misfolded proteins. It is also effective removal of terminally damaged polypeptides and induces extra cytoplasmic stress response. It plays a major role as a master protease which is attached to different substrate by different specificity factors. The main in case of protein A, it consists of 202 amino acids with tripeptide linker and hexahistidine tag, where the propeptide is cleaved. In case of mutant, it is composed of 215, which includes sequence of 14 amino acid propeptide, and it has a molecular weight of 24135.65 and the number of amino acids being 215 and pi is 6.04. The main difference is in the catalytic trade. The in the wild type, the nucleophile is being serine, and but the in mutant type, it is being replaced with alanine. So it loses its activity, but forms heterocomplex, complex, but won't be able to digest protein like wild type because it loses proteolytic activity. So it can't cleave its peptide. Thus, it is composed of 215 amino acids, while wild type undergoes autocatalysis and during maturation loses its propeptide sequence, forming 202 amino acids.
So the purpose of the entire experiment is to identify a given unknown protein based on its size and sequence along with the determination of purity, concentration and stability. So we started with size determination following STS-based technique that allows protein separation by mass using polyacrylamide gel as support medium and sodium dodecyl sulfate for denaturing protein, while concentration is determined using Beer-Lambert law. Then we perform the sample for mascot analysis where we expect to observe the peptides matching with sequences in dataset done by washing, dehydrating and digesting the gel slice extracted from STS. Then we performed immobilized metal affinity chromatography for purification of protein from bacterial lysates. So in this case we used E. coli as our host cell for protein expression where induction of protein was done using IPTZ. Our protein has hexahistidine tag that has affinity with nickel beads. So we use this technique to separate our protein where the bound proteins will be eluted with high concentration of imidazole which has structural similarity with histidine. During the elution, our protein sample might contain chemicals like imidazole sodium chloride which interacts with BCA reagent while performing BCA assays. So buffer exchange was done in order to separate the interacting buffer components from protein. In BCA assay, addition of BCA reagent to protein sample generates purple color by chelation of cuprous ion and the absorbance is measured at 562 nanometer. And finally, for determining the thermal stability of the protein, thermal shift assay is performed where real-time PCR machine is used that changes the temperature while measuring fluorescence. And finally, we were assessing the activity of the protease enzyme that releases the coumarin and leads to fluorescence, which was earlier quenched.
Since we knew our sample clippy proteins were engineered with hexahistidine tag, we purified our protein by IMAC technique. Protein were induced by IPTZ, which can be observed from a STS cell image where there is high relative abundance of protein band around the zone of 20 to 25 kilodalton. On subsequent elution with imidazole, we can observe purified band of protein A and B. However, few contaminants can be observed in protein A and molecular marker lane, which might be due to the overflow of sample B while loading it into the well. After purification process, we determine the concentration of both proteins by BCA assay, where a standard curve was plotted by measuring the absorbance of bovine serum albumin diluted in different concentrations. Based on graph and the absorbance value of test protein, concentration of wild-type ClP was calculated to be 1625 microgram per ml, while the concentration of mutant ClP was calculated to be 512.78 microgram per ml, which was comparatively low, which might be due to the loss of sample during the buffer exchange process. After the concentration determination, 5 microgram of protein was electrophoresed to assess its purity. So observing the image, we can say that there is possibility of contaminant in protein A. However, equal relative abundance of proteins in both the lanes suggests that concentration might have been determined accurately. While doing PTS assay to measure thermostability, curve for protein A was similar to positive control, but the curve for mutant ClP showed number of peaks, which indicates the possibility of presence of other species in sample with similar melting temperature. The background fluorescence of positive control is quite high, which indicates the presence of some hydrophobic surface residues or partial denaturation of protein in the sample. The melting temperature was predicted to be around 73 degrees Celsius for wild type and 69 degrees Celsius for the mutant form. So the mutation might have slightly lowered the thermostability of protein. Peptidase assay was done to determine the activity of proteins. Wild-type proteins showed protease activity, which can be observed by increase in fluorescence in proportion to its concentration. However, wild-type protein did not have activity as much as positive control. This might be due to the high purity of positive control compared to the sample. While we plotted the graph for mutant ClP, fluorescence was not observed at all, so it doesn't show any proteolytic activity. From the experiments, we can conclude that we were able to purify our protein from bacterial lysate by IMAC. Concentrations were accurately determined by BCA assay and purity was assessed. Regarding the effect of mutation, there was not much difference in thermostability, but the activity was completely absent in mutant protein. Regarding future direction, ClP protein has been target of new classes of antibiotics, where its activation can degrade bacterial proteins in human body. One study showed acyl depsipeptides can be used to activate ClP, so the experiments can be designed to assess the activity of wild-type ClP in the presence of acyl depsipeptides.